Welcome everybody to our non-live stream journal club. Due to technical issues with the internet, paper we're doing is our third journal club. It's targeting endogenous proteins for degradation through the Affinity-Directed Protein Missile System by Fulcher et al. And it's basically a technical paper on a new fusion protein that's been developed to bind to proteins and then target them for destruction. And that's basically what most of molecular biology and biochemistry is about, right? It's all about things transiently binding to things, other things, and then those things doing things, right? So the whole paper is going to try to convince you that this thing binds to another thing, and then something happens, and that something is degradation of the target. So how do they do this? And what is this Affinity-Directed Protein Missile System, or as they their acronym states, AdPROM. Well, let me share my screen and let's take a look at some of the components of the AdPROM system. So here's the paper, and I'm going to scroll through the paper, and they have a little cartoon here of their complex here in the figure 1B. You notice that it's got this, kind of complicated thing, this monster with Col2, RBX1, E2. The actual AdPROM is this, these two boxes that say VHL and nanobody, monobody. And these are the two parts of the AdPROM system. The nanobody, monobody is kind of the seq part, which binds to your endogenous protein target. And the VHL part, which comes from this protein called, I think, von Hippel-Lindau syndrome protein. This is your destroy part. So essentially what this system does is one half of this system binds to your target protein. The other half of the system binds to a complex of other proteins that are within the cell. And this complex basically attaches these little protein groups called ubiquitins onto target proteins, and these ubiquitins, when they basically bind in a certain array or configuration, they actually direct this protein to be shunted off into the proteasome, which then causes it to be shredded, right? So the two parts of this, the one part that's actually variable is this nanobody, monobody part, right? The VHL is constant. And by altering the nanobodies and monobodies, you can get this system to theoretically bind to whatever you want it to bind. So what are these nanobodies, monobodies? So my screen is still being shared, I guess. So antibodies, most of you probably have heard of them. Most of you probably worked with, some of you have worked with them. You probably know what they do, but just a refresher. The antibodies that we are familiar with, they're basically this Y-shaped structure. It has a heavy chain and a light chain. And at the very business end, at the very tip of these two arms of the Y, you have a variable heavy chain and variable light chain. And these two in conjunction bind to, at the very tip, bind to antigens, right? So your immune system churns out billions of variations of these, and that's how they can basically bind to things they haven't seen before. So anyway, these antibodies are huge in biotechnology. I think about 50% of new cancer drugs, anti-cancer drugs, rely on some derivative of an antibody. For example, Herceptin is the kind of trade name for an antibody binding to the HER2 receptor, which is a receptor tyrosine kinase that's upregulated in certain breast cancers, right? So that's a big target right there. And in their paper, they're actually also going to be targeting proteins that were within the RTK, or receptor tyrosine kinase pathway, because it's medically relevant. A lot of these signaling pathways, especially the RTK pathways, are upregulated or mutated or overactivated, overstimulated in cancers, causing the cells to basically uncontrollably proliferate. So instead of using these variable regions from regular antibodies, they're using these variable regions, which are single domains, from camel antibodies. So camel antibodies are different. They're simpler. They have basically two heavy chains, no light chains, and they have just one domain, one variable domain. So it's a lot smaller than your fragment antigen binding domain. So Smaller is better when it comes to packaging things into vectors, which you need to introduce it into cells. So that's one main reason to use it. So that's the camel-derived antibody, the monobody. What they also utilize in their system are, well, actually, no, that's not called a monobody. It's single domain camel antibody. Monobody is actually very similar to the single domain from the variable region, but it's not from an antibody. So here's a picture of a beta-pleated sheet that's the domain of an antibody. And this red part here is the variable region that'll bind to antigens. It's actually, let's go back here. It's actually a domain from this protein called fibronectin, which has a very similar structure. And it's also small, very small. So scientists have been able to mutate all these residues and basically have it act like an antibody or well, they call it monobody, right? That was a term that was coined. So this part is the guidance system, right? So that's going to cause this whole complex with the VHL part that recruits the whole ubiquitination part for targeted destruction for the proteasome, right? So here's just a kind of an overall image of just a very rough image of the ubiquitination pathway for destruction. Ubiquitin, I know, is used for all sorts of other signaling events, but in this pathway, it's when certain lysine residues are covalently bonded to these other residues, they tell the cell to start chewing up and destroying. Okay, so that's a little background. So what did they do in the paper? What did they target it against? Well, they're targeting against two proteins, but the first one they're looking at is this SHP2 protein, or SCHP2 protein. So this protein is downstream of a signaling pathway from a receptor, either an RTK, or I think it's also involved in these JAK-STAT pathways. So essentially, it's a sort of a chain, like a bucket brigade of proteins that will phosphorylate other proteins, and all the way at the bottom, you have transcriptional factors that turn on other genes, and those genes regulate the growth cycle of cells, right? So a lot of these components, like RAS, for example, are found mutated in many cancers. So they're clearly important targets for anti-cancer therapeutics. So for their proof of concept, they're using SHP2, and they're actually testing their AdPROM system in several different cancer cell lines, a whole bunch of different ones, to show that it's kind of not cell line dependent so much. So one more little bit of background is there's another system that's very similar to their system called PROTAC, and I'm just mentioning it because they mention it and they say their system's easier to use. And PROTAC functions very similar to their system in that it has two ends. One end binds to the target protein, so it could be a monobody. The other end is an E3 ligase recognition domain, which means it will bring forth this complex that will then cause ubiquitination of a target protein and destruction. So I believe, if I'm not mistaken, that the way these two domains are linked is actually through a chemical linker that needs to be added via a series of chemical reactions. So this isn't just a fusion protein where you can just code it and have it expressed as one protein, right? So there's, I think, some extra steps involved in preparing it. So that might be a bit daunting for some labs, right? So anyway, back to the paper. So let's close this. Let's go back here and let's see, stop share. Back to my face. All right. So Steve, you got any questions? There's no audience. So was that clear fairly as a background?
This is the part of the show where you'd like me to say, oh, actually, yes, we've got a question from our audience from Facebook, but we actually haven't.
Yeah, maybe you can make one up. Anyway, I can go back to the paper. All right, let me go back.
Sven's got anything to say.
Yeah, all right.
Any observations?
This was just background. Anyway, so share screen. Okay, back to the paper. So what do they do? So we'll just jump right into the results. First of all, there are a lot of Western blots in this paper, In fact, it's all Western blots. You know, I'm just going to explain a little bit about what a Western blot is, because you have a lot of bands all over the place. And basically, it's an old method that's still relevant and used to identify specific proteins in a cell. And cells obviously have thousands of different proteins, tens of thousands, you know, even more if you count all the variants that are modified temporarily, like phosphorylated, ubiquitinated, whatever, farnesylated. So what you do is you essentially get a cell extract, and when you run your proteins on a gel, you've basically got everything in there, right? So if you were to stain this gel, you'd have bands all over the place. So if you want to identify your particular protein, you have to then transfer your gel. You have to basically make a print onto filter paper. And we could talk about the technical ways to do it some other time. But once you make that print, you can then use specific antibodies. So this IB on the side of this blot here is, I believe, an immunoblot, what it's immunoblotted with, right, or against. And the antibodies are usually conjugated to some sort of, you know, some sort of reporter. Could be an enzyme that, you know, when you develop this, it'll leave, you know, a colored precipitate. There's a million things, well, not a million, but you can attach all kinds of things, fluorescent molecules, whatever. So the intensity of band, just like in a regular agarose gel for nucleic acids, you know, correlates with the amount of protein. So the darker the band, the more the protein. And the position of this band correlates with the mass of the protein, more or less, right? So on the left, you've got your mass in kilodaltons, molecular weight, 75 kilodaltons down to 10 kilodaltons. And on your right, you have what this blot has been probed for. You can strip a blot and reprobe it over and over again to a certain extent. And I'm just going to mention the three things that they're going to probe for. So, okay. So they're introducing a couple of different things into the cell. One is obviously their VHL nanobody monobody, right? Which is directed against two domains. They have two different ones, and one they call ANSA1, the other ACS3. Those are the monobodies, and they're targeted to two domains, NSH2, CSH2, within the SHP2 protein, right? So just two different sites on the same protein, and these ANSA1, ACS3 are basically the monobodies. VHL ANSA1 and VHL ACS3 are these monobodies conjugated to that protein that recruits the whole proteasome complex, right? So these monobodies are the search and the VHL is the destroy. So for controls, they just have the VHL domain alone, which is just the destroy domain, but no search. And then they have the two monobodies without the search and destroy domain. So those should not affect the target, you know, to any great extent as far as degradation is concerned. And then wild type has nothing added into it. And over each of these gels, so they're figure 1, C, D, E, and F, they're basically looking at 4 different cell lines: U2OS, MDA, MB468, A549. You could look all these up if they interest you. And they're basically just testing a whole bunch of cell lines to show that, you know, this is broadly applicable. Right. So let's just, as an example, take a look at U2OS. We don't need to go through all of them. So on the blot, what we see is you have your antibody against SCHP2, and that's going to tell you the amount of this protein that's endogenous that they're targeting for destruction. So you notice in wild type, you got a lot, but when you add the VHL domain or the two monobodies without any VHL conjugated, you have a nice thick blot, the band, so it's not being destroyed. And lo and behold, when you add these VHL conjugated monobodies, you see a dramatic decrease in amounts of SHP2. So it appears to be working. All the way on the bottom, you see this immunoblot GAPDH. That's just a loading control. You usually pick some protein that's not affected by what you're doing, so that way you can show people that this decrease is not because you just didn't load sufficient quantities of protein, right? And then we have this IB flag, and this IB flag is against these monobodies. And in order to see these monobodies, they didn't have an antibody against ANSA1 or ACS3, but they had this little tiny protein called the flag tag attached to it, which doesn't interfere with the activity of the protein, but you can generate an antibody against it. It just makes things, makes things easier because there's already commercially available antibodies for it. So this just shows you that that the ANSA1, the VHL, the ACS3, and the VHL-CS3 are being expressed in the cell, right? They don't have the VHL tag, so you're not going to pick that up. And you notice there's size differences. ANSA1 is small, and the ACS3 monobody is small, so they're hovering between 10 and 15 kilodaltons. When it's conjugated to VHL, the whole whole fusion protein is about 37 kilodaltons. So that is figure 1C. And you pretty much more or less, to varying degrees, see similar things for all the other cell lines, right? Maybe slight variations in, amount of degradation of SHP2, but you see the same theme, right? So the take-home message of this is proteolytic AdPROM degrades endogenous SHP2 in multiple human cancer cells, as the figure legend states. All right, so I'm going to stop screen sharing for a little moment here. Is my face back on? Yes? All right. So I guess I've been chided that I should have a little bit more face time, go back and forth. That's somehow like more natural than me, just like a disembodied voice. All right, so I'm still here. And I guess we'll go back to the paper. So that was figure 1. Let's share screen.
Yes, apparently people enjoy seeing you, Oliver. You should be flattered.
Yeah, I am. I don't particularly enjoy seeing myself, but
who does? I mean, same for me. I mean, I hate listening to myself on recordings.
Yeah. Okay. So yeah, and then notice that some of these gels have ERK1-2. That's just another loading control. Later, we're going to look at ERK2. It's actually downstream, and it gets affected by SHP2, but it's the phosphorylation status. So let's go down. Let's take a look at figure 2. So this figure here is basically trying to demonstrate that it is this proteasome-mediated, E2 ligase, E3 protein, pathway that actually targets SHP2 for destruction. And they want to demonstrate this by using a chemical, MLN4924, and they treat the cells for various hours. And this chemical, what does it bind? It's a place called P-SHP2. Forgot exactly what it binds to. Let me find it in this paper. Okay, with around, what exactly does MLN294 bind to? Okay. So this this degradation happens through this COL2CRLR machinery, and for this to happen, there's a protein that's, I guess, called NED that needs to attach to another protein, and it's called NEDylation. So that needs to happen for this whole cascade to happen. So this compound inhibits that, right? So you can block the proteasomal degradation. Figure 2, okay. So figure 2A, again, you got your controls. Here they just picked U2OS cells to look at. You have wild type, ACS3 only, which is a negative control, and VLACS3, which is your actual seek and destroy system. You have SHP2, and then you have GAPDH, and then you have this other protein, HIF1A, which is this protein, I believe, is it's another pathway that's affected by SHP2. We can go back to that later, but it's being used as a control essentially here. So wild type, you notice that if you block MLN 4924. Nothing is getting degraded or nothing is, being this is the baseline, right? Because you're not doing anything. Also with the ACS3 only. Now the VHL ACS3 with 0 minutes treatment of MLN2924, there is no SHP2, so it's been degraded. After a period of time of incubation with this inhibitor, you notice that the SHP2 levels go back up. So it appears that it is dependent via this chemical on this proteasomal degradation pathway. What do they do in figure B? Precipitation. Oh, here they're doing a binding assay. So this is different from these Western blots in that instead of loading all of the cell extract and taking a look, trying to find your protein, you're actually testing for interactions, right? What binds to what? And then you use basically an antibody that's attached to these little beads, and it's called also immunoprecipitation or a pull down assay where you essentially precipitate two things that bind. So if one thing binds to another thing, they're going to come out as a complex, and if one of those things is bound to something heavy, it's going to precipitate out of the solution. Then you can get rid of everything else. So what are they looking at here? So they're doing an immunoprecipitation against flag, and the flag tag is attached to the CS3, which is also found alone. That's the monobody, or also in VHL ACS3. And then they're immunoblotting against SHP2. So that just shows you here that if you look at up on this immunoblotting, after they've pulled it out, you notice that with this MLN2924, it's either they added or not, it's irrelevant. You still have the ACS3 binding, so this chemical doesn't affect the binding of this monobody. It'll still bind and pull out your SHP2. But with VHL ACS3, you notice you have a little bit of pull down, right, when you block it with MLN294, but most of it's degraded. So I'm not sure why it's only been able to pull down a little bit if you've blocked most of the degradation. Because if we look here in the SHP2 input, now input is basically everything that's not immunoprecipitated, right? So that's basically the band up here is what's coming out of here from the input. So there's a lot of SHP2 in these cells that have been blocked with MLN4924, but just a tiny bit coming out with VHL ACS3. So, maybe the SHP2 is binding to other things. I'm not sure. But so these monobodies are specific and they bind to SHP2. So then later they go to figure 3. And they're basically here looking at downstream events, right? So they're showing that when they get rid of SHP2, you have all sorts of things being altered that are downstream of the pathway, right? There's these proteins ERK1/2 that affect transcription, and they are phosphorylated. So these pathways usually phosphorylate things one after the other. And I'm not going to go through all of this, but we'll just take a look at figure 3A. And these are just different cells. They're looking at MDA, MB468, K562. Might take a look at figure C. So for figure A, they're stimulating the cells with epidermal growth factor, EGF, right? So you've got 0, 15 minutes, 30 minutes, 60 minutes, 120 minutes of 50 nanograms per mil EGF. And they're looking in the cells. They're doing a Western blot and seeing what happens, right? So the IBs on the right side, you've got anti-SHP2, then you have anti-SHP2 Y580. Now the Y is just a shorthand for tyrosine. So a lot of these kinases, they're either serine, threonine, or tyrosine, kinases. So Y580 is just the tyrosine at position 580 that gets phosphorylated. And you can come up with an antibody that's against a phosphorylated version of SHP2. Same thing with ERK1/2. You've got two phosphorylated residues on ERK2, threonine and tyrosine residues. Then you have regular ERK2 and GAPDH. So those are loading controls. Notice they're all kind of the same amount across. Wild type, you keep adding EGF. The ship levels actually look like they go slightly down, but you can see that the phosphorylation goes up, right? So by adding in EGF, the phosphorylation levels go up. When you add in the, so this is downstream, right? So the SHP2 phosphorylation and also the ERK2 phosphorylation goes up. Oh, sorry, that was, the SHP2 phosphorylation goes up and the ERK1/2 phosphorylation goes up. When they do ACS3, the SHP2 phosphorylation basically goes away. And it could be that ACS3 is just blocking SHP2, right? Because SHP2 is here, but the phosphorylated variant isn't. So it might be just, as a monobody blocking phosphorylation. And VHL ACS3 degrades SHP2. Obviously, you don't see phosphorylation of SHP2 anymore because it's gone. You might see there's a slight attenuation of ERK phosphorylation. You do see it going down, right, after so many minutes. The same thing with ACS3. So there's obviously other things that are phosphorylating ERK1/2, but it could also partially be SHP2 mediated as well. Okay. So in figure 3C, they're looking at basically signaling downstream of this protein that's found mutated, very famous protein called BCR-ABL. It's found mutated in a form of leukemia, and it's famous in that one of the first designer type drugs was developed against it that specifically targeted BCR-ABL. It was called here they call it imatinib, and it's a small molecule. It's also known as Gleevec, and it's had tremendous success at, I think it's CML, chronic myelogenous leukemia. That's the condition, and imatinib is prescribed for that. There are issues with it in that cells can actually mutate their BCR-ABL, so imatinib is not functional anymore, so they have other variants. So they are now looking to see what happens downstream of this BCR-ABL signaling pathway, and if targeting SHP2 will have any downstream effects, similar to imatinib, right? So here's wild type. You have plus, minus. You have, this is a very busy blot here. So you either have no BCR-ABL. So this BCR-ABL is actually expressed in these cells, just FYI, I think it's expressed. C, right. So D is cells, these K562 cells, they have natural C-ABL expressed. So this one here just has some kind of vector attached. So either they have BCR-ABL expressed or they don't have it expressed. And then you either add imatinib with BCR-ABL expressed, or there's no imatinib. So C-ABL, right, immunoblot. So no BCR-ABL added, obviously there's none. You add it, there it is. Add BCR-ABL, SHP2 is highly phosphorylated, and then it goes down when you add imatinib. And if we go to just the ACS3, which is our controls, you notice SHP2 is still phosphorylated, but when you add the VHL ACS3, SHP2 is gone, and also obviously the phosphorylated variant is gone. So if we look at what was downstream before, and there's additional proteins that are down 3. This is the STAT proteins. I'm going to skip over ERK1/2, and I'm going to just go to STAT proteins. STAT3 alpha is not affected. PSTAT3 alpha, which is the tyrosine phosphorylated version. Wild type, you add BCR-ABL, it's heavily phosphorylated. You add imatinib, it's blocked. And you see the same thing actually with adding in well, also the monobodies. Some blockage appears to be actually, let me see if I'm actually reading that right now. Wait. ANSN4. So with just the, so without the imatinib, right, you don't have any phosphorylation of the STAT proteins, but you do have phosphorylation if you express BCR-ABL. Actually, let's see, BCR-ABL. And adding in imatinib is going to decrease phosphorylation. So it could be that a little rusty on my downstream components for the STAT signaling, but it could mean that this imatinib is maybe affecting other components and not just SHP2. So SHP2 isn't the sole thing that's responsible for STAT phosphorylation. We could review that back later. What else do we have here? Okay. Figure 4. So figure 4, they look at more downstream signaling components. And I'm being told here by my, my timer that I have 7 minutes remaining on my freebie here. So I'm going to apologize. I'm going to skip through figure 4. There's more blots. They're actually compare this system in figure 5 against another system that's used to knock down proteins called siRNA or short interfering RNA. The take-home message from this is that VHL ACS3 works faster. So it takes 48 hours to knock down with siRNA, and it takes 24 hours with VHL ACS3. And 6, they just look at a different protein, endogenous ASC. So that's another protein, and they show it's not just working against the SHP2 protein. Anyway, their system appears to work. Before we have a discussion, they do mention that there are 2 problems with this system, and they both are, not knocking the system, because they both, are kind of outside of the scope of this. One is just the delivery, right? You still have to get the system into cells. You still have to put it encoded in a vector, and they mention that in the paper that that's a limiting step, right? So if you want to target some protein somewhere and it's behind the blood-brain barrier, well, you still have that problem to kind of overcome, right? So this is just the system that does the job. It's like CRISPR-Cas9, right? CRISPR does its thing, but you still need to deliver it someplace. The other issue is that this targets proteins for proteolytic degradation. And I know that for us, we're all interested in aging, right? And a lot of these proteins, or a lot of these compounds that build up in cells are, defective proteins, misfolded proteins, aggregates of proteins. And the problem is that I don't think a lot of these are, the cell has problems digesting or destroying or proteolyzing, right? So how efficient would this system be if, let's say, we had a monobody against some sort of misfolded protein or beta amyloid plaques or something like that? Would it be able to, probably not proteins that are, or probably not plaques or things that are actually like neurofibrillary tangles that are outside of the cells, but proteins that are misfolded inside of the cells. How effective would this system be? Because it's utilizing the endogenous proteolysis system of the cell, which has problems digesting these things. So it would be really interesting to see, if by kind of selectively targeting these misfolded proteins, can you kind of, by, pushing them, giving it a nudge, can you get the proteolytic machinery to degrade those? I don't know. It would be an interesting experiment, I think, to see how effective this would be against, I would think that there would be pretty recalcitrant, but, maybe just by increasing the dosage of, bringing them closer to the proteasome, that might force it to eat them. I don't know. So that's kind of like my main, that's what I'm curious about, right? And actually, if that's not the case, then perhaps you can make a similar system where it's tethered maybe to an enzyme that can destroy or degrade these misfolded proteins, right? So instead of having it bound to VHL, you'll have this bound to, I don't know, some archaebacteria enzyme that will, chop up these misfolded proteins. I don't know. I'm just throwing that out there. Anyway, any questions in the remaining time that we have? We have 3 minutes before we have shutdown. Let me stop screen sharing here. Oops.
Well, as you mentioned, currently it needs to be imported using a vector and expressed in the cell. But maybe we can take this system and fuse it to a cell-penetrating peptide like HIV-TAT. Or maybe rather than using the monobody or the antibody, we could use an alpha body, which is already cell-penetrating, as I understand it.
Yeah, so those are ways to overcome that limitation. Yeah. So it'd be interesting to see what kind of variations on this can be derived. Again, sort of like obviously for cancer research, you want to target these proteins that are hyperactive or overactive because that's what they're focusing on. But for aging research, these misfolded proteins, that would be really neat to see if you can get these misfolded proteins gotten rid of. Is the system good enough to do that, or does it need to be, tethered to something else? And if you get rid of these misfolded proteins, are they the primary causative agent of cells going senescent and, aging? So what will happen to these cells after you introduce this? So that's what I'd be interested in seeing.
It's a good question, and hallmarks of aging certainly suggests that loss of proteostasis is one of the primary reasons we age. And I was going to talk about senolytics, but not in 1 minute, 26 seconds. I'm not going to. And I was going to mention BCLW as a possible target for cancer and senolytics. I don't know. Did you want to relaunch it? Stop the recording, relaunch it, and then finish? And have a quick discussion.
Yeah, sure. Okay. So
Keith's going to be pissed that we didn't use the paid account. He's going to edit this bit out, by the way. Keith, this bit where I swear, edit this out, all right? Sorry about that, those important messages.
Actually, we never went away after the splicing. We're talking about seeing how well, I mentioned seeing how well the system works against really effed up proteins that are like, misfolded, that are oxidized to varying degrees, that are aggregates, and how well would it, because the cell has problems already dealing with this stuff, with its native systems of proteasomes, would it be sufficient just to bring these, these really badly performing proteins that have problems being digested in the first place to the proteasome? Or maybe we could use, you need to conjugate the antibodies to some other enzymes that would do the job.
Yeah, assuming that they can't be digested.
Yeah.
Or is it a case that the cellular machinery just isn't as efficient as it used to be and it just builds up? Is there too much,
Well, if you talk about protein aggregates, then the problem, if you have intracellular aggregates, is going to be probably that they just are like large clumps of protein, and the proteasome just cannot digest that. It needs to take a protein and unfold it and feed it into the proteasome for degradation. So that's going to be the problem here. It's probably just like the proteasome cannot even physically start digesting this. So what you probably will need is a type of system that breaks up these clumps into individual misfolded proteins that can then be degraded.
Yeah.
Like you have amyloid inhibitors that do that. How is that dye called again that does that? You have a, I forget. It actually extended lifespan in C. elegans. You have a, yeah. Okay, I forgot the name.
Yeah, I don't know. I mean, you could certainly maybe tether the monobodies to something else, maybe like chaperone proteins or something that can help unfold and maybe make it more edible. I don't know. There's a lot of things. I mean, the most baseline experiment would just be to see how well this system works against misfolded proteins. The prediction is not very well, but who knows? Maybe just increasing the local concentration of the proteasome might be enough to tip it.
Theoflavin T, by the way, was what I was mentioning.
Excuse me?
Theoflavin T is the name of the dye I was mentioning.
Oh, theoflavin T, okay.
It actually extends the lifespan of C. elegans by inhibiting the aggregation of amyloid proteins.
I love all these silly names that they invent. Good, isn't it? Sonic the Hedgehog's my favourite, but that's a gene. And the other thing I was thinking of it could be potentially used for is senescent cell removal, because it could be things like the BCL-W,
right?
BCL-W, BCL-XL, and BCL2. So it's the BCL2 family. Those are shown by Campisi and others to be essentially pathways that allow the senescent cells to resist apoptosis. So, if you can actually degrade the proteins that they use, could potentially use it for senolytics, I would think.
Yeah, but I agree. But you'd still have to get over that other issue of the delivery, right? You want to gotta specifically get it to the senescent cell and not start apoptosing all your other stuff.
Well, obviously, yeah. I mean, we don't want mast cell death, because that would be bad, right?
Is there a good senescent cell surface marker that's out there?
No, not really, as far as I know.
When the usual way of detecting them, it's not a very good one, is beta-gal, isn't it?
Well, it's beta-gal, and also what's that other protein that's overexpressed?
P16.
Yeah, P16. Yeah.
Well, P16 ink, isn't it?
Yeah. Right.
Yeah,
those are on the
interior. Ideal though, because senescent cells are not the only cells that express them. There's some concern that stem cells, when they're dormant, also express these things. And in fact, Alexandra mentioned it, if you remember from the panel we had, that her concern was that senolytics may affect stem cells adversely.
Yeah.
Yes and no. I don't think it does, because if it did, we wouldn't see life extension, and I don't think we would see the positive health benefits. So I'm going to go out on a limb and say I think it's a warranted concern. We've obviously got to test it, and we are at the MMTP. But I don't think it's necessarily the case. Otherwise, the mice would definitely die a lot earlier, wouldn't they?
Yeah. So in this case,
stem cells out. Yeah.
So again, this kind of sort of alleviates the delivery, but not so much, because now you can target your AdPROM could be turned on by promoter that's responsive to P16. So if it gets into the other wrong cells and they don't have enough P16, it's hopefully not going to be turned on, but you got a lot of cells to target. So then, how much vector are we talking about that you're infusing, right? So, it's like real estate, right? The three most important things are location, location, location. And for these therapies, the three most important things are delivery, delivery, delivery. So
remember that now. That's going to be the catchphrase, isn't it? And
right. I mean, there's a lot of things that'll kill cells, but getting it to that specific cell is a problem.
Well, it's like people, I've heard said, yeah, well, a 45 caliber bullet kills cancer. Mm-hmm. It's a bit crude, but yeah,
it'll work.
The researchers were talking about application for cancer, I believe, and Alzheimer's in the articles, and So, and also one of the things that they were talking about was it would have potential to be able to help treat the undruggable.
Who was that?
Because they're saying that there are certain target proteins that are not affected by small molecules, and AdPROM opens up the opportunity for them to actually target those molecules. They're proteins, sorry, molecules. So literally they're saying that it could allow us to target the undruggable. I don't know what you thought about that.
Yeah, so speaking about the undruggable, there's one thing I forgot to mention in the paper, and that was actually something I skipped over before we got kind of pulled. That was, let's see where we go, ERK2. This was this small molecule that they used, SHP099, in the paper. That was it was a small molecule inhibitor of SHP2. And the interesting thing is, let's see if I could pull this back up. All right, so I'm gonna just pull this on my screen. Now we got a little bit more time. We can go back. Where is this darn thing? Okay, figure 4. So they're looking at different cell types, and I've got figure 4 up. And when they add in SHP099, different micromolar amounts, downstream components are phosphorylation of PERK1,2. And if you increase the amounts of this chemical that's an inhibitor of SHP2, you notice that you have a pretty dramatic decrease in phosphorylation of PERK2. Some of these, it actually affects the amounts of SHP2 as well. Actually, no, that was actually the basal level is pretty low in these. Sorry, I didn't look at the control. So you have a decrease in phosphorylation that's, pretty substantial. And this is supposed to be, I believe, specific for SHP099. But if we look at the experiments done with your AdPROM protein here, let me see a good example I can get. For some of these cells, MDA MB468, so VHL-ACS3. So we're going to look at PERK1,2 phosphorylation. So VHL-ACS3, just remember, it totally destroys SHP2. There's like no SHP2 present, right? Like gone, destroyed. You add in, so it's kind of blank. You add in EGF and stimulate it, and you then get phosphorylation of ERK1,2. If you add in SHP099, with or without EGF stimulation, you have no phosphorylation of ERK2. So kind of the take-home message is that this phosphorylation of ERK2, which is downstream of SHP099, could be, it could be affected by other. So one interpretation could be that the chemical SHP099 actually not only inhibits SHP2, as it was kind of initially, I guess, screened for, but other targets as well, right? So kind of might explain why you have a much more thorough knockdown of phosphorylation of ERK1,2. And if truly, this VHL system is very, very specific for one protein, although, you'd have to check every protein to see if that were the case, then that might be an interesting way to validate whether the specificity of your small molecule, right? Because they should act the same, right? But if you target SHP2 and you destroy it, and you have some phosphorylation go away because it's downstream, but you get all of it go away with this SHP099, which targets SHP2, then it's not fully SHP2 dependent phosphorylation. There might be other side pathways, other kinases, and your SHP099 has cross-reactivity. So anyway, I just wanted to point that out.
So, can we use it for aging or not? That's probably the question that the audience wants to know. Is it a definitely maybe? Are we going to commit to a definitely maybe?
The safe answer is always, I don't know.
Or it's plausible.
Yeah, it's plausible. I mean, it's another thing. It's dependent on how important these misfolded proteins are and whether this system can be employed somehow to target those proteins, right? So if there are certain proteins that are bad or certain compositions of macromolecules that are bad that are correlated with aging, like directly correlated, and this could be used to target them, then yes, it could be used for anti-aging. So those are a lot of assumptions and ifs, right? So if that and that and that, then yes.
Yeah, and don't worry, we're not going to sue you if it turns out that we can't.
Yeah.
Well, it seems this system is very likely useful against cancer, and cancer will be one of the main issues we have to overcome.
Sure.
In that sense, it's likely if it keeps working.
Yeah. I mean, you have to realize this system is, the strength of this system, like CRISPR, is that it's got broad applications, right? You could use it to target a lot of things. So it's not specifically applicable just to aging, but like you mentioned, cancer and probably a whole bunch of slew of other, diseases that have bad proteins that are bad actors or some other macromolecule, right?
But cancer's a good thing, right? I mean, it's like I'm saying. I say quite often to people, is if we're gonna solve aging, if you'll pardon the phrase, then we need to solve cancer anyway, because cancer is. I mean, I certainly, from my point of view, would say that cancer is the poster child of aging, as far as I'm concerned, because it's mostly to do with genomic instability.
Sure.
So cancer kind of is aging, really. So even if the mainstream researchers who are not focused on aging are still working on cancer, they might not understand the relevance to aging the way we do, but it's still a good thing. And at the end of the day, as I said to you earlier today, if people develop the tools, they might not be thinking of the same things as us, but we can still use those tools.
Yeah. We certainly can.
So basically, we can all become immortal space ninja goblins. That is still the plan, right?
Well, as long as we get the delivery problem solved.
Exactly.
So maybe in our future papers, we'll talk about delivery systems.
Yeah, it's not a bad idea. I mean, and I know you wanted to delve sort of more into the
theoretical stuff,
into the theoretical side, and I think, to be honest with you, I think people would enjoy that, because we have been pretty hardcore, haven't we, for the last 3 episodes?
Well, we've been focusing more on kind of the engineering of solutions, right? So maybe focus back again on the kind of inherent problems and theories of aging and the data that's being collected that suggests aging could be this or that, right? The causative agents. It certainly seems to be an accumulation of bad stuff, right? And it might vary between organism and organism. So it'd be interesting to check out some papers on that topic.
You know which one I'm going to mention, don't you?
The one on Hydra?
Well, that, I mean, as aging theories go, you know what my favourite is?
Do I?
Should do it.
What do you say?
Ben knows. I'm always talking about it. I'm on commission.
On what? What?
Hallmarks of aging.
Oh, but come on, that's a lot of different things. Come on.
But you know that, I mean, the actual paper itself is, as far as I'm concerned, a pretty good view of what aging is. I mean, okay, I mean, SENS as well, of course, is great. Slightly different categories, but essentially that's the same thing. And the approach is repair.
Hmm.
So, you know, yeah, we can talk about that. You know, why not?
Why not? Indeed.
We can talk about your favourite 80s movies if you like, Oliver.
Okay.
It's up to you.
Okay. Well, I'm gonna just shut off the recording now. I think we've explored these issues as far as we want to explore them for this journal club.
And for the record, it's Big Trouble in Little China. Just saying. That's a good movie, you
It's
know?
a great movie. Yeah.
Did you know that they're making another one?
They should not.